Let me introduce the last speaker of this morning. This first session is Eva Voss, who's going to talk about photoreactivity, nucleobases, ancestors. Eva, are you there?
Yeah.
I see you. Can you share your screen, please?
Yeah. Can you see it?
Yes. Perfect.
Okay.
So 20 minutes.
Thank you. So thank you so much for giving me the opportunity to present my work. And it's a bit different for everyone, the rest of the works, because, well, it's more applied work instead of developing or implemented code. So, well, it is well known the important role that DNA plays in life, and precisely because of that, the chemical, biological, and physical properties of DNA has been studied for decades. And in fact, the structure of DNA and RNA is completely determinate, and 5 are the canonical nucleobases can be considered essential for life. But we have a question at this point is why this particular nucleobases, why no others? And in order to find an answer, we have to go back to the very beginning. Along the earliest period, primary chemical compounds under string condition of temperature, UV light, or reduction atmosphere gave place to the first organic compounds, among them nucleic acids and proteins. The nucleic acid and proteins were able to create a strong association between them, generating the first protocells, which millions of thousands of years later evolved to the current organisms, I mean, superior organisms. Simultaneously to this biological evolution, a chemical evolution must have taken place. An incredible amount of compounds can be considered to take part of the genetic material, but instead, just 5 of them compose our alphabet, our current alphabet, genetic alphabet. Well, it's in the biological case, chemical evolution was also ruled by the survival of the fittest, and knowing the selection processes to which the nucleobases were suggested, we can understand better the final decision of the nature. And UVA light exposition must have been a relevant selection factor, as it doesn't seem casual that all the canonical nucleobases present a clear photostability upon UVA absorption. And the deactivation mechanism of these canonical nucleobases has been studied, and the presence of descending slopes and the presence of conical intersection between the lowest single excited state and the ground state allow the biomolecules returning to the ground state in an ultra-fast time scale. So at this point, the question will be, what generates this photostability? And attending to the structure of these biomolecules, just few options can be explored. And for example, it is the aromatic ring the main responsible for the photostability, or is really necessary the functionalization of it? Moreover, the position of these substituent groups have any kind of consequence. So several theoretical and experimental works could help to understand this evolution. And not purine and neither pyrimidine core are the main responsible of the photostability. In fact, once the biomolecules are photoactivated, the generation of log-line excited states occur and triggers a secondary reaction along the excited states. So discarding the aromatic ring as the responsible of the photostability, the functionalization of them should be a determinant factor. And with that premise, the photophysics of the oxo-modified pyrimidine and purine nucleobases has been examined, And particularly in this talk, I'm going to present the results obtained for the OXO modified at C4 position in pyrimidine core, the 4-pyrimidine. So the main factors of the potential energy surface of the excited states has been mapped, extended multi-state sp2 level, using an active space of 12 electrons and 9 orbitals. And in this way, we have optimized a stationary point as minimum transition states or degeneracy points. In order to connect different regions of the PES, we have compute MEP, NEP, and LIP calculations at the same level of theory. And finally, in order to determine the probability of population transfer between excited states of different multiplicity, spinor coupling have also computed with OpenMolCAS code. So with the entire static picture, we can find possible and accessible decay mechanisms that the system can use in order to dissipate the excess of energy. Moreover, dynamic analysis was also performed in gas phase and with the same level, extended multi-state sp2, and consider in the same way that the static analysis, singlet and also triple excited states. For this purpose, we have create a Hoehnc interface that combine 3 different softwares. The first, a version of Shark, which is in charge of propagating the dynamics under surface hopping approach. Then Bagel to compute the analytical extended multi-state sp2 gradients. And finally, OpenMolCAS to evaluate the probability of hopping between singlet and also singlet and triplet excited states. So, well, the complete study was also carried out in close collaboration with the experimental group of Professor Carlos Crespo in Cleveland. And starting with the static picture of the 4-pyrimidine, at the ground state, the equilibrium geometry is characterized by the planarity. From this point, vertical excitation energy has been calculated, and they are indicate that there is only one bright excited state, the lowest single excited state, with a PP star character. So for the Frank Condon ray, the system can be photoactivated, reaching the S1 excited states. From here, we expected that the system evolved to a more stable situation, and minimum energy path calculation conclude in the first PP star minimum, which this minimum, on contrary with to the Frank Condon region, is characterized by the puckering of the C2 atom. And near closing energy and similar, geometrically similar, we found another minimum. In fact, these 2 minima are separated by a small energy barrier. So in order to discover or to find a deactivation mechanism, we searched conical intersection between S1 and ground state. And 2 S1, S0 conical intersection were located, which are very close in energy to the S1 minima, really similar in geometry. In fact, these conical intersections are directly connected with the minima. So minimum energy path from these points along a ground state concludes that the system could return to the Frank Condon region and getting a complete deactivation. So this pathway is very probable. However, we were interested in explore the triple excited states, and in fact, the lowest triple excited state at this region of the potential energy space is really near in energy. And for example, the conical intersection, in fact, there are triplet degeneracy points. And the SOC value calculate suggests also a possibility to population transfer from S1 to T1. From this point, along the lowest triple excited state, the system could evolve to a new PP star minima in the T1 excited state, which recovers the planarity of the Frank-Condon region. And at this point, well, we didn't find any ground state T1 conical intersection, intersystem crossing point. So from here, we expect that the system deactivate by a radioactive process. So, well, this proposed deactivation mechanism has been corroborated by experimental results, and the translation absorption spectra show a main band at 320 nanometers with a broad tail extended at 700 nanometers. This signal uniformly decays along time until completely disappear after 30 picoseconds in acetonitrile and less than 5 picoseconds in water solution. So these results indicate not only that the 4-pyrimidine is a photostable system, but also show that activation occurs in an ultrafast time scale. In fact, these times are comparable with the canonical and, well, with the fastest canonical and no canonical nucleobases as adenine or hypoxanthine. So in order to study the evolution of the excited states a long time, we have carried out not diabatic molecular dynamics. So a total of 100 trajectories in gas phase at extended multi-stage gas PD2 have simulated during 500 femtoseconds. And firstly, the system is photoactivated to S1. Well, most of the system is photoactivated to S1 in agreement with the vertical excitation presented previously. And during the simulation, most of the trajectories return to the ground state. And just less than 30% continues in the excited states, mainly in the S1 stage in black. Just very small fraction of the trajectories are able to populate the triple excited states. So this, with the no experimental evidence of the population of the triplets, indicates that this path is residual. So now focus on the deactivated trajectories. We analyze in detail these trajectories, and we saw that most of the trajectories present a interconic, suffer internal conversion processes governed by a conical intersection that characterized by the C2 distortion, coinciding with the conical intersection found in the static picture. With these trajectories, we could estimate the time life that is, agrees with the experimental time constant that is in the same order of magnitude. So the dynamical results agrees with the experimental and also the static results, which indicates that the 4-pyrimidine is a photostable system with a ultra-fast deactivation mechanism. However, we look out in more detail the deactive trajectories, and we found interesting and unexpected behavior. So some of the trajectories that get the ground state follow a different and complete different mechanisms and doesn't lead the original and planar structure, and instead the opening of the ring occur. 23% of the deactivation trajectories suffer this dissociative reaction. And surprisingly, these trajectories don't present any significant difference from the photostable trajectories. And the internal conversion funnel is also characterized by the hooking of the C2 atom. So, but in contrast, when the trajectories get the ground state, the ring open, generating the ketene photoproduct. So this ketene photoproduct, well, we identified two different conforms, the cis-ketene and the trans-ketene. And in order to understand better this photodegradation reaction, we perform a net calculation between the conical intersection and these two ketene species. And the optimized reaction path so that so the necessity to return to the original ground state equilibrium geometry before getting the opening structure. But this path need to surmount an incredible energy barrier. So this is in disagreement with the dynamical results, in which we saw that from the conical intersection, the generation of the ketene occurs directly. So we think that this photodegradation mechanism is governed by the dynamic effects. In fact, if we analyze the momentum at the moment of the jump, we observe that velocity is accumulated in this interesting bond, but in different direction, provoking the opening of the ring. Fortunately, these results are corroborated by experimentalists, and steady state absorption spectra detect significant changes in the absorbance along time. So this suggests, well, this indicates that the new species are generated on function of time. In fact, 3 different species of photoproduct were separated, and with the experimental absorption spectra, we are able to identify them. So the red signal is associated to the compound, to the parent compounds for pyrimidine, and the other 2, the experimental absorption spectra has been compared with simulated spectra of potential photoproducts. And we conclude that the black signal can be associated with the carboxyl acid, which is the product of the hydrolysis of the ketene that we obtained in the dynamics. And the other, the blue signal, can be attributed to the hydrated species of the 4-pyrimidine. So in conclusion, the incorporation of the oxo group at the C4 position of the pyrimidine core promotes the photostability of the compounds. And this incorporation alters the photophysics of the excited states, activating the efficient internal conversion funnels that allow a fast deactivation mechanism. However, this oxo group doesn't guarantee the chemical integrity of the compounds because of the photodegradation reactions. And at this point, well, it seems necessary the double oxo substitution in natural nucleobases, as in uracil, to preserve the integrity of the genetic material. And Well, thank you so much for your attention. And if I have time, I would like to discuss with you why finally I use Bagel program instead of OpenMolcast.
Thank you very much. Okay, so some questions from the audience. I see 2 questions in the chat. So Felix again, could you explain your dynamics approach? Why is it not possible to compute hopping probabilities with Shark plus Bagel?
Well, in fact, Bagel doesn't have this kind of calculation. I mean, he is not able to compute SOC, for example. So in order to consider this population transfer, we need OpenMolcast. Well, as I already understand it yesterday, I think Shark right now have this kind of implementation, but I'm not sure.
Okay, Federica also has another question. Have you done studies with different time steps as QS switches depend on the time step?
No, in fact, no. I'm sorry, I cannot answer this question. I mean, the time steps that I use for the dynamic is 0.5 femtoseconds.
Okay. Perhaps was tested in other studies before.
Yeah.
Okay. Other questions from the audience? Okay, if not, let's thank Eva and all the speakers of the session again.